Hello everyone, and this is my presentation on meiosis. And if you don't know what meiosis is, it's the splitting up of your cells to make more cells. And that means you're going to go from one cell to four cells in meiosis. And also in meiosis, it has two parts, one being meiosis 1 and one being meiosis 2. Now, in meiosis 1, we are going to start with prophase 1. And in prophase 1, this nuclear membrane represented by purple wool is going to get broken up. And once it's all broken up, like so, these centrosomes are going to move to opposite poles, and these microtubule spindles, as shown in glowstone, and the orange wool are the centrosomes. But these microtubule spindles, they're going to form around these chromosomes shown here. Now, these chromosomes are from the father and the mother. And as you can see here, 2n equals 4, that means 4 chromosomes. And 2n means it's a diploid cell, which means it's diploid meaning 2. So there are going to be 2 cells that form after this. And that means that n is equal to 2, and you will see why in a second. But the second part of meiosis 1 is metaphase 1. And in metaphase 1, meta meaning middle, the chromosomes are going to line up at the center of the cell. And these microtubule spindles are both going to attach to the chromosomes and to each other, meaning this centrosome will attach its microspindules to this centrosome, and it doesn't have to hit the chromosomes as seen on this outer one, this middle one, and that outer one. And once it does that, we move on to anaphase 1. And in anaphase 1, it's actually going to split up the chromosomes, and these microtubules, they don't shatter like that. These microtubule spindles are going to pull onto the chromosomes and pull them to each pole. And these microtubule spindles that are only connected to the other centrosome are expanding the cell to make room. And after anaphase 1, we move on to telophase 1 and cytokinesis. And in this, the one cell is going to form into 2 cells. And in both of these, the chromosomes that were split apart are now going to have the nucleus reform around the chromosomes. And this will happen in both new cells, like so. And the reason why n is equal to 2 is that, as you can see, 2n meaning diploid cell, which means it has technically 2 cells in it. There's 4 chromosomes in this one, and in the single chromosomes that were split up, there are 2 chromosomes. In each cell, there are going to be 2 chromosomes for meiosis 1. And now the same thing will happen with both sister cells that were formed from the one big cell. And this will happen in meiosis 2, which we will start with prophase 2, just like we started with prophase 1. And here the same thing happens. The centrosomes will move to opposite poles. This nuclear membrane will break up in both cells. Like so. Those centrosomes will move to opposite poles as well. And we will move on to metaphase 2, where the chromosomes will now line up on the middle of the cell, and the microtubule spindles will attach to the chromosomes and start to pull them apart in anaphase 2. Now we had chromosomes. We started with chromosomes. Now we are seeing sister chromatids, meaning those are going to get split apart. As you can see, there's two sister chromatids. And those will get split apart from each other in anaphase 2. And we now move on to our final part in telophase 2, where those two cells will now split up in half again, and they will form 4 cells, as I said in the beginning. And the nucleus will reform around the sister chromatids. And once they do, that will be the end of meiosis. And something I did not go over that could happen is we could have a phenomenon known as crossing over, which means either in prophase 1, when the chromosomes link up with each other, they could cross over like so. We could have one chromosome going here and then just cross over and come this way, and the same thing would happen with the mom's chromosome, where it will also cross over and it will form an X like this, and that's where we could have some father chromosome and father chromatid in there and some mother in the same cell in telophase 2. However, in this scenario, it did not happen. And that is it. So a quick rundown. In prophase 1, the father and mother chromosomes are going to link up, and microtubules are going to attach to centrosomes, and those centrosomes are going to move to opposite poles. In metaphase 1, the chromosomes are going to form at the middle, and the microtubule spindles are going to attach to them. And in anaphase 1, those chromosomes are going to be pulled and split apart by the microtubule spindles. And in telophase 1 in cytokinesis, those centrosomes are going to go away, and a nuclear membrane is going to reform around the chromosomes. And that same exact thing will happen in meiosis 2 with prophase, nucleus splitting apart, centrosomes moving to opposite poles, right there and there. In metaphase 2, those chromosomes are going to form at the middle of the cell. Anaphase 2, those are going to now get split apart, and they are now sister chromatids that are being split apart, not chromosomes. That is a very important thing to remember, is that in anaphase 2, it is sister chromatids that are being split apart. In anaphase 1, it will be the actual chromosomes that are being split apart. And in telophase 2, the nuclear envelope, as you could say, or nuclear membrane will form around each sister chromatid to form 4 new cells. And this yellow glass is just the cell membrane itself. And that is meiosis. Hope you guys enjoyed this presentation on it, and I will see you in class to learn more about biology.